There are 30 million people in the world living with HIV, and although we have good treatments, we still don't have a cure. The reason we don't have a cure is because of a small quirk of the HIV life cycle. Normally, HIV infects cells, inserts its DNA into the DNA of the infected cell, then makes more virus right away. Very rarely, a cell infected with HIV won't start making more virus right away. These types of cells are called latent cells, and they're the reason we don't have a cure. The problem with latent cells is that they're ticking time bombs. They look just like all the other T cells in your immune system, but they have the ability to make more HIV virus at any time. These latent cells are also created within 2 or 3 days after infection. Researchers at Rockefeller University wanted to learn more about these latent cells to find ways of picking them out of the crowd. To pick out and study latent cells, they created a new latent cell capture technique called LURE. To capture latent cells with LURE, they take blood from HIV-positive patients on antiretroviral therapy and activate any of the latent T cells that were present in the blood with specialized molecules. They then recover any T cells that were activated and analyze them to find out what makes them so special. 2 major findings came from analyzing the activated T cells. The first is that the latent cells they collected all seem to come from a single cell becoming latent and then dividing to create multiple latent cells, rather than multiple T cells becoming infected and then becoming latent independently from each other. The second major finding was that the latent T cells have different levels of active genes than their non-infected counterparts. The genes that have higher levels of activation in the latent cells are genes that shut down the virus and allow for cell division without making new HIV viruses. This research is just the beginning of learning about latent cells, and it only focused on cells that circulate in the blood. Plus, the cells were only taken from 3 different patients. More research is needed to see if these insights hold true for other patients and other sources of T cells. Even with these caveats, we're still learning more about latent cells than we ever have. It's possible that this research may tell us the secret to picking out latent cells, allowing us to turn our current treatments into cures.